Researchers at MIT's Little Devices Lab have developed a set of modular blocks that can be put together in different ways to produce diagnostic devices. These plug-and-play devices, which require little expertise to assemble, can test blood glucose levels in diabetic patients or detect viral infection, among other functions. Using this system called Ampli Blocks, the MIT team is working on devices to detect cancer as well as Zika virus and other infectious diseases. The blocks are inexpensive, costing about 6 cents for 4 blocks, and they do not require refrigerator or special handling, making them appealing for use in the developing world. Over the past decade, many researchers have been working on small portable diagnostic devices based on chemical reactions that occur on paper strips. Many of these tests make use of lateral flow technology, which is the same approach used in home pregnancy tests. Despite these efforts, such tests have not been widely deployed. One obstacle is that many of these devices are not designed with large-scale manufacturability in mind. Another is that companies may not be interested in mass-producing a diagnostic for a disease that doesn't affect a large number of people. The Little Devices Lab researchers realized that they could get these diagnostics into the hands of many more people if they created a kit of modular components that can be put together to generate exactly what the user needs. To that end, they have created about 40 different building blocks that lab workers around the world could easily assemble on their own, just as people began assembling their own radios and other electronic devices from the commercially available electronic breadboards in the 1970s. In this case, the components consist of a sheet of paper or glass fiber sandwiched between a plastic or metal block and a glass cover. The blocks, which are about half an inch on each edge, can snap together along any edge. Some of the blocks contain channels for samples to flow straight through. Some have turns, and some can receive a sample from a pipette or mix multiple reagents together. The blocks can also perform different biochemical functions. Many contain antibodies that can detect a specific molecule in a blood or urine sample. Those antibodies are attached to nanoparticles that change color when the target molecule is present, indicating a positive result. These blocks can be aligned in different ways, allowing the user to create diagnostics based on one reaction or a series of reactions. In one example, the researchers combined blocks that detect 3 different molecules to create a test for isonicotin acid, which can reveal whether tuberculosis patients are taking their medication. The blocks are color-coded by function, making it easier to assemble pre-designed devices using instructions that the researchers plan to put online. They also hope that users will develop and contribute their own specifications to the online guide. The researchers also showed that in some ways these blocks can outperform previous versions of paper diagnostic devices. For example, they found that they could run a sample back and forth over a test strip multiple times, enhancing the signal. This could make it easier to get reliable results from urine and saliva samples, which are usually more dilute than blood samples but are easier to obtain from patients. The researchers are now investigating large-scale manufacturing techniques, and they hope to launch a company to manufacture and distribute the kits around the world.